virus, unfortunately, partly maybe because of the haste and partly because many of them were actually developed in not certified laboratories and academic institutions. Many of them, unfortunately, give false positive or false negative results. So that made actually the detection and the containment of the virus also more difficult. So far, the virus had actually a few mutations. The reason I am saying it a few, because viruses are usually mutating a lot. Some of them mutate some of them do that really a lot, and some of them do it less so. So actually, the coronavirus has some stability, and that is a little bit of encouragement so it's not mutating very frequently and very fast. There was a very encouraging data early on before the actual vaccine development started in patients, that non-human primates could not be reinfected after they recovered from the coronavirus, which was actually a very encouraging sign when we started the vaccine development. So I am showing you this slide to give you a feel that usually how long does it take, because you hear this in the news, that this vaccine development happened extremely fast and then people are questioning the validity of that. It is true, it happened extremely fast, and then I will explain to you why. So these are actually the general phases of development for any drug. They start with research, then they go through an early development, which includes the phase 1 and the phase 2A clinical trials. The phase 1 trials are always on healthy volunteers, and these are actually intended to investigate only the safety of the drug. And starting the phase 2As when actually people are going to be treated with disease. And the phase 2A trials are supposed to prove that the drug is actually working to treat the intended disease. And then the phase 2B and phase 3 trials then go into a really large number of patients. And then after the trials are finished, then they need to be submitted for evaluation and hopefully acceptance to health authorities all over the world. And then once it is accepted and approved, that it can be lodged into the public. Now, if you look at the timing of all these different development phases, the research and the early development on the average, and especially when you are talking about small molecule development is always taking much longer than large molecule development, and vaccines are large molecules. So large molecules are usually a little bit faster. But in small molecule development, the research until you prove that the drug is actually going to work in patients, it could take about 6.5 years. And it costs about $650 million. But once it actually does work in patients, and you actually have to really put it into a lot more patients in order to determine true safety and efficacy, that could take another 7 years. And it could cost between $1 to $7 billion. And then once you go through the submission and the launch time, normally it also would take about at least a year or a year and a half, depending on if it is like a fast track compound, like many times for cancer development, for instance, then it could take another 1 or 1.5 years. So this is actually indeed a very long process. And the reason why it is a long process, because you take one step at a time, you evaluate your results, and then move to the next, and the next, and the next. And you don't do anything in parallel, and you don't take any risks. You just sequentially, very pragmatically, go from the beginning till the end, and that's why it's taking so long. So when you look at the vaccine development phase, then again, these are the same phases of development. In this slide, it also says that from identifying the agent until you actually get to this proof of concept stage, it could take 1 to 10 years. And then the phase 2, phase 3 can take another 2 to 7 years, and then the approval process in another year. In case of the COVID-19 vaccine, we actually got into this phase 1, then we actually start the clinical trials in 8 months. And the reason for that is because we were able to sequence the compound very quickly. As soon as the sequence was made, my company at least knew what kind of construct they actually want to use to design the vaccine itself. And the reason why we knew that, because we already used that contract in many other vaccines. So we have used the same contract for the SARS vaccine, for the MERS vaccine, for Ebola, which is already approved in Europe and is going to be filed in the United States, and also the HIV vaccine, which is currently under investigation, which means that this construct that we have used have been actually treated over several 100,000 patients over many, many years. And that gave us a lot of confidence that this construct is actually very safe and it really can be used in humans. And that is the reason why also it could cut back on the vaccine development time a lot. And so we were able to actually create these constructs. We were able to test them in non-human primates very quickly. And we were able to determine their safety in these models. And that's why we were able to actually move into the first in human and the human clinical trials much faster than we normally would. Now, the other thing is that when the compounds or a vaccine, for instance, is being developed, they at first develop the vaccine, they actually make enough of that particular one that it's enough for the clinical trials. They finish all the clinical trials, so we are up to about like 10 or more years of development. And once they are absolutely sure, then they start manufacturing it. And that's when they make, enough doses that they can treat the general public later, which will take also some time, maybe another year or 2 or 3. In this case, as soon as we actually had like 3 or 4 different viable constructs that they may actually work at the end and we can put them into clinical trials, we immediately started to manufacture them in large quantities. And as soon it was clear that which one of those 3 is actually going to be the winner, then we immediately made a lot more other contracts with a lot more other manufacturing companies. And at risk, even at the start of the clinical trials, we actually started to manufacture really large amounts of doses of the vaccine, which is completely at risk. And it costs a huge amount of money. And we were really hoping that at the end, it's actually going to work. And that's the only way how you can actually save time. And this is the only way how you can actually raise the time in order to develop the vaccine. So again, in this slide, it is just summarizes what Joseph said. So we worked very closely with the Food and Drug Administration and also with BARDA, which is the Biomedical Advanced Research and Development Authority in the United States, in order to develop the vaccine very quickly. We did a lot of non-clinical and clinical testing and the manufacturing process parallel at the same time, rather than waiting for the results of each of them and do them sequentially. The virus vector is well known, as I said. We used it for Zika, HIV, and RSV vaccines as well, and tested them. We had a very high level of confidence about safety. The scaling and the manufacturing was also done at risk in parallel, as I said. There was a large collaboration between many different manufacturing companies. And we also can cut down the approval process, because in this approval process, the health authorities are actually going to approve these vaccines, and approved already several of them, for emergency use, which means that they are having a much more expedited review and approval process, and they are making the approval based on the interim data, whatever the companies provide. But the clinical trials are actually also going to continue for further safety and efficacy of each of these vaccines. So my company actually also promised that by the end of the year, hopefully we will be able to present 1 billion doses for the use. So how these COVID-19 vaccines cause the disease? So they actually enter into our lungs through mouth, nose, eye droplets, which is containing the virus. So it's all in aerosols. That's why it is so important when you go to the dentist, like for instance, most recently they changed all of their processes which would actually create aerosols in the office, like when you used to do like cleaning with water or polishing and things like that. They don't do that anymore. They do it all manually in order to reduce the aerosols that could be in the air and potentially could spread any viruses, not just this one, but anyway. So what this virus does is it enters the body through a specific receptor, which we actually have in our lungs, the adenine converting enzyme 2 receptor, which is ACE2 receptor. Some of you who may have heart disease or high blood pressure, you may take ACE inhibitors in order to prevent that. And that's the receptor that this particular virus is actually uses to enter into the body. Why is it important? You also heard that children are less susceptible to this virus than elderly people. And the reason for that is that children have much fewer number of ACE receptors than elderly people. As we age, our ACE receptor number is actually increasing. You also heard that the people who have heart disease, there might be more susceptible to the virus. Again, because it is likely that they may take actually ACE inhibitors in order to reduce high blood pressure. And ACE inhibitors also can increase further these ACE receptor numbers in the lungs, which again increases the likelihood and the susceptibility for the virus to enter the body even easier.
Ilkiko,
So,
what is an ACE receptor? What does it do? What's the purpose of it?
It is basically helping to convert the adenine in your body. So it's just a receptor which is related to the work and the metabolism, etc. of the adenine.
So why do old people have it in greater numbers?
Well, many of our receptors actually, that as we age, some of them just increase in size. I mean, it has a general biological, I don't really know the answer for that. I mean, there are a bunch of other receptors also that may increase as you age, and some others decrease. So it just depends.
Thank you.
It just happens to be one of those that increases.
How does it overwork the immune system? Normally, our white count is about 7,500 plus or minus 1,500. And when we have an infection, then it will zoom up to 20,000, 30,000. So is it producing too many white cells to react to it, or just how does it overwork the immune system?
Yeah, I will show you it in the next slide.
Thank you.
So and then again, there are a number of different. There are basically two major viruses. The viruses even have RNAs at their contact or DNA as their contact. Viruses themselves are really tiny things which are sort of in the liminal space between life and living things and not living things. So a virus itself, it either has an RNA or a piece of DNA, and it has a bunch of protein coat or a coverage around it, and that's basically it. It's a very, very tiny thing. And when it is on itself and it's sitting on the surface, it's basically not living. But as soon as it gets into an organism, then it basically uses the organism's machinery and all of the biological machinery in the organism's body to replicate itself. And then once it replicates itself, and once it enters into the body, it will actually evoke an immune response because of the protein that the virus has will naturally evoke an immune response in the system. And once this immune response is actually becomes really, really large, because you have a lot of virus entering into the body and using up your machinery to create itself and replicate itself, it actually also releases a lot of additional cytokines and other immunological substances in your body that actually some of them become extremely toxic. And those immune responses actually cascade and start a cascade that is going to increase inflammation And I will show you in the next slide. And in this particular case, all that inflammation is mostly focused on the lungs. And once these inflammations in the lungs become really severe, and we actually create a lot of fluid and liquid inside the alveoles of the lungs, that is basically what becomes completely detrimental. So the virus, this coronavirus, is actually an RNA type of virus. So once it enters into the cells, it releases the RNA content, and it uses the cell's machinery to synthesize its own protein, and it assembles into the new virus. The new virus is affecting a new cell, and it's just keep replicating itself if we don't stop that. It stimulates the immune response, it overworks the entire immune system, and it creates very severe inflammation into the body. And that's basically what becomes the issue. So in this picture, you can see that more. So this is the virus. Inside of it, this is the RNA. And then around it is basically this protein coating, which has these little spikes that they always show on the pictures. So it basically enters the body, it releases this RNA content, and as I said, it uses the machinery of our own body to replicate itself many times. And in the meantime, it also creates an immune response. And some of the virus is going to be actually taken up by certain immune cells and killed. But unfortunately, if there are too many viruses coming at us and into the system, then after a while, the body just cannot really deal with that anymore, and it really increases inflammation. And in this picture, it particularly shows that the inflammation is actually happening in the lungs. These are the ends of all of the different processes within the lung called alveoles. And in these alveolar systems, once the virus really enters into the cells and increases the inflammation, then the fluid level really increases in all these alveoles, and that really creates the shortness of breath, and ultimately that people will not be able to breathe anymore. And that's unfortunately leads basically to death. So in order to stop this pandemic, what we need to do is that we actually need to reach herd immunity. And what is herd immunity? It is when 70% of the population will be protected from the infection. And there is only two ways that you can reach that protection. Either that somebody gets sick and then they will develop their own antibodies against the pathogen, and that's how they will become protected. Or if somebody will get vaccinated, and that way will raise the antibodies against the pathogen. So the only way to get immune against the pathogen is to have enough antibodies against it in our bodies.
Is one way better than another, or more effective than another?
I'm sorry?
Antibodies that your body produces yourself, are they more effective than the vaccine?
So the vaccine also is not an antibody. The vaccine is actually, I will show you, the vaccine is actually going to trigger in your system to create the antibody.
Okay, so
the antibody is you producing. Okay. Now, you can use antibody for the treatment of the disease when somebody is very ill. That's what President Trump got, and some other people. And actually, just about a week ago, one company, Eli Lilly, was approved to treat patients with their antibody, which they create and synthesize, and they actually basically add it into the patient. So that can be done as well. But that is basically really helping the most severe patients and prevent basically death at that point. But naturally, the best thing is if you raise your own antibodies against the pathogen, because then you will be protected from it for a long time come.
Okay, thank you.
Ilkiko?
Yeah.
Can I ask a question? I had a friend say that so when you get COVID and you have antibodies versus getting the vaccine, aren't you protected longer with the vaccine, right?
So that is unfortunately not totally clear for us, because we haven't been able to run these clinical trials long enough to be able to answer that question. I mean, the hope is that hopefully, some of these vaccines indeed may have longer protection. for everybody, but we won't know until they actually will run the course. So these trials really need to go on for several years to be able to answer that question. And as you remember, there are a bunch of other vaccines that you also need to get boosters of. Like the tetanus vaccine needs to be boosted after 10 years. The flu vaccine needs to be given every year. You know, if you get the shingle vaccine, you need to get a booster 4 months later. So, I mean, it really just depends. I mean, each vaccine is different, each pathogen is different. It just really depends.
So then people who have had COVID, should they get the vaccine anyway?
So there is no indication that they could not. So I have read about that, whether you know it would be any detriment if they would get the vaccine. No, it isn't. It is okay to get that. And again, the reaction what you are going to get into the vaccine very much depends on also what was the severity of the infection. How many viruses actually infected you? So it is possible that you have a very mild disease. You got just really a few viruses, you know, who infected you, and then you have a healthy immune system, and you are able to generate enough antibodies that protect your body from it. And you know, your cells are going to remember that antibody. So when you get infected again by chance, then those antibodies are going to be created much faster probably, and will going to make a better protection. But you know, the level of antibody in your body when you get like a mild case of the disease may not be as high as what you would get if you will actually get a well titrated number of viruses through the vaccination process.
Julie, I think the CDC, this is Teresa, the CDC is recommending that people who have had COVID go ahead and get vaccinated.
Yeah, yeah. So there is no counter indication against it for sure. Yeah.
Thank you.
So this slide is also again just shows that there is a number of different kinds of vaccine that you can develop against any pathogen. And for this one, we also have all over the world a number of different kinds of vaccines that people tried. They can use the inactivated virus itself to create a vaccine. The best example for that is like the flu or the polio vaccine. They can create just weakened viruses, like for instance, it is the case for the smallpox or the chickenpox. They can create the vector-based viruses like the Johnson & Johnson virus, for instance. And then you can create RNA or DNA-based viruses. In this case, it's RNA-based viruses, what is the Pfizer and the Moderna virus in this case. And every country in many different pharmaceutical and biotechnology companies, they really are working still in this moment on a number of different types of viruses. It is a good thing actually to have them in our toolbox, because different people will react differently to different kind of constructs. All these viruses, I mean, all these vaccines, sorry, have also a number of other ingredients in them, all sorts of excipients and other components. And some people may be allergic to certain components. And so one vaccine maybe has that component, another vaccine doesn't. So it's actually a good thing to have a number of different kinds of vaccines available for the general public. And now I am going to try to show you that video. I hope it is going to work. Let me find that. Where did I put the... okay.
One type of non-replicating viral vector vaccine is called an Adenovector vaccine, which uses an adenovirus as a vector. Adenovirus causes the common cold, but the vector is genetically changed in the vaccine, so it cannot replicate. Once the Adenovector vaccine is injected into the body, the immune system starts making antibodies. These specific antibodies work to attack the piece of the coronavirus. Then, once the perceived threat is gone, the immune system creates memory cells. Memory cells remember how to fight the virus. So if and when they encounter it again, the immune system is ready to fight it off. This means the body is immune. One type of non-replicating viral vector vaccine is called
And that's it. I think it's kind of short and sweet. If you would like to watch it again and again, I am happy to share with you. I think it is actually very informative because it really visually shows, how actually the vaccine works. So let me see if I can.
It would be nice if there were human actors playing the antibodies and the virus.
Hold on. I have to go back to my presentation.
Who said that?
Oh, just a lurker.
That's Richard. I recognize the voice.
All right, and then this last slide, I just wanted to show you again, the many different companies, and this is obviously not complete, but there are many, many different companies who actually started to work either on the virus vaccine, or on treatment for the disease. And so what we have approved so far is the BioNTech and Pfizer vaccine, which is already, as you know, used in the United States. Also for Moderna, it's a misspelling, actually it's Moderna. I also mentioned Eli Lilly that has an antibody treatment that has been approved, I believe, like 1 or 2 weeks ago. And also there is an AstraZeneca vaccine in collaboration with the Oxford University that was approved in Europe and being used in the UK and other places. But they just filed also in the United States. So probably it will be approved as also in the United States. And the Johnson & Johnson vaccine, so we actually released the phase 3 data on this vaccine that is also filed for approval in the United States for the emergency use application. It is going to go into the advisory board on February 26th. And the advisory board, in case of Moderna and Pfizer, really did the approval in just a few days. Usually the FDA follows the recommendation of the advisory board. So hopefully we are going to also gain approval relatively shortly. And as soon as the approval is reached, then we will be able to start providing the available stockpile of the vaccine, which is already available. The trucks are ready, almost running, to be able to take that to the different distribution sites.
Ilkiko, I'm sorry to interrupt. And you may not know the answer to this, but do you know how it's decided which vaccine goes to which part of the population?
So there is basically the task force, the coronavirus task force in this country, and the World Health Organization, and every country has their own task force basically, and they are making the decisions that where the distribution is going to be happening, what are the different cohorts, in what kind of sequence are going to get vaccinated, etc.
Ilkiko, what drives the different companies to pursue one type of vaccine over another? What criteria make them decide that they're going to go for an RNA DNA-based one? Or?
Yeah, that's a very good question. I mean, it partly depends on their own technology, right? So what is the technology that they already have and available? For some of them, it's really the length of the development. So for instance, in case of BioNTech and Moderna, they actually had the technology and they knew that they are able to do this really fast. You can see that it was much faster than creating an adenovirus construct vaccine. But like the downside is that you actually needed, this construct is actually less stable. That's why you need to have -80 degrees centigrade to store them, and or at least the Pfizer one. And also they are not strong enough to invoke an immune response. So they actually need to get them in 2 doses in order to have a booster shot. And for this, our company basically decided that we already had indeed, again, the technology. We had a lot of data on this construct, so we were very confident about its safety. We knew it's going to take longer to develop. But at the same time, we were hoping that you will be able to give only 1 shot, which is the case. And the construct itself is much more stable. So it's actually stable in regular refrigerators, I think up to 3 months, and in freezers for up to 2 years. And so that makes the distribution and the inoculation much easier.
Thank you. Yeah.
2 questions. One's a follow-up to my previous question about white count. When a person is infected, how high does it run up the white count from 7,500 to what? 20,000, 30,000?
Yeah, well, I am not a medical doctor. Unfortunately, I will not be able to answer that, but maybe somebody else who is a doctor can.
Okay, so second question on, correct me if I'm wrong, but my understanding is that the Johnson application is effective about 65% versus the Pfizer and the Moderna 90-some% with two shots. If somebody had their preference, if they could select one, what would be the preferences given a 65% probability with 1 shot versus 95% probability given 2 shots? So
right. So so let me say that that those percentages are extremely misleading. Whenever you are actually comparing them, you are really truly compare apples to oranges. The trials that Pfizer and Moderna run were extremely different than the trials that Johnson & Johnson run. We actually run the trials against a number of different mutations. The Pfizer and the Moderna trials were done only on the first kind of vaccine, or the first, not vaccine, sorry, the first kind of virus mutation. Also, the data that you are seeing here is an interim data. These trials are actually going to run much longer, and the number of incidences you run the trial longer is going to also increase. And the treatment factor is going to increase. So as you increase the trial length and your vaccine is actually working very well, those percentages are going to increase as the trial goes on. So I really would not hang up on those percentage numbers. They actually really meaningless. Right now, the vaccine development is really happening in a fishbowl because of the political situation. So we have to release all sorts of data and information, what normally actually the general public would not see. And it's very difficult to compare all these different things. And I wouldn't hang up on that. If the vaccine works, if it is approved, it is actually creating enough antibody in your system, then you should get it, doesn't matter which one. If you are not allergic, for instance, to the Pfizer or the Moderna vaccine, people have to know that they are pegylated vaccines, so they have polyethylene glycol in them. Some people are allergic to polyethylene glycol and they will have an anaphylactic shock. So those people cannot get those vaccines. They have to wait for another one that is going to be approved. But if you are not allergic and you can take the, I mean, if the Pfizer vaccine is going to be available for me the first time, that's what I'm going to get.
thank you.
Is it true that one vaccine prevents serious disease as opposed to, I mean, I had heard somewhere, and once again, I have no idea if it's true, that one vaccine actually, even though the efficacy is not as high a percentage, which I know we don't need to pay attention to, but that the efficacy for preventing truly serious disease and death is much higher. Is that true?
Exactly.
Or
well, so the way how the trials are designed, then before you actually do your trial, you have to design what are the endpoints that you are going to measure at the end of the trial. And different companies may have different endpoints they measure. So you can measure outcome from mild disease or severe disease or death. Again, the Johnson & Johnson trials were actually measuring also the endpoint of really severe diseases and death. So it really depends what was the original protocol for the trial, what were the endpoints they were measuring it against, and then if that endpoint was, for instance, if your endpoint is that you wanted to treat mild disease, to have 95% on that is not very difficult to reach. But if your endpoint is that you actually want to prevent death completely, then it's probably a higher hurdle. But for the Johnson & Johnson vaccine, actually, it was very effective to prevent hospitalization and death.
On the concept
Can I just a second? Do we have a couple more slides that you
No,
want to show
this
us?
is it.
No,
This is
if not, would it be okay if we stop share screen so we could just see each other so we could all sort of talk? It's hard to do it on the, thank you.
My question would be on the death rate, 1 in 52. Is that a high death rate for a virus?
So, I mean, each virus had very different, I don't know the exact statistics on all those. I can tell you that the death rate, for instance, of the 1918-1919 influenza was much higher than the coronavirus death rate right now. And one of the differences is actually the healthcare system. So the death rate very much also depends on the healthcare system. And the death rate might be very different in the United States, for instance, or in another country, depending on how they treat their patients.
And the stem cell, how is the stem cell studies that are just going on incredibly fast right now, how are they used with making a virus?
So stem cells are not, I don't think that stem cells are really used making any virus. Viruses are naturally occurring, they are not being made. You maybe wanted to say if stem cells are being used to make vaccines.
Yes.
Yeah, okay. So stem cells are not really being used to make vaccines. Stem cells might be able to use to actually treat any sort of illness or diseases where you actually have to regenerate certain cells. With respect to virology and for the treating viruses, that's probably not would be the first line of treatment.
Okay, thank you.
Ilkiko, I have a very self-interested question, and I had no idea. I wish I had known this earlier that you work for Janssen. You and I are in effect colleagues. On November 16th, I was given a double-blind placebo trial for the Janssen vaccine. And on December 16th, I came down with coronavirus. Now, that means and I was fairly certain I was given the placebo because I had absolutely no reaction to the injection. Mm-hmm. I had no vax hangover. I had no irritation at the injection point. Having said that, either I am if I in fact did not get the vaccine, if I got the placebo, I just proved that I'm a vulnerable member of the population. If I did get the vaccine, one, I think that I probably am costing you some money, and two, I think that I'm not at all certain I want to get another vaccine again, having now had the virus and now having naturally recovered and recovered fairly quickly with a mild. I was asymptomatic by about 10 days, and I felt kind of draggy for about a month later.
Your code hasn't been broken yet, has it?
What's that?
Your code has not been broken yet.
No, and that's actually what I wanted to ask you. And I'd asked the medical research because obviously Janssen, like all the big pharmas, can't do the research, can't do the protocols themselves. They have to have independent third parties doing them. And I've asked my research center that, when will my trial be unblinded? and they won't tell me?
At the end of the study, most likely if you were in the first cohort, that it's actually running for 2 years. So naturally you are going to be unblinded after 2 years.
So I'll never know. I will not know.
Well, but you know, the thing is,
whether or not I got it.
yeah, I know, but I mean, the thing is that, you know, what your participation in the trial is incredibly valuable, and I cannot thank you enough.
Well, thank you.
To volunteer for it and help us with all the data, because whatever is the data, you are contributing to that, and it's extremely important.
I am wildly pro-vaccine. However, I do think it's reasonable if in fact a actual vaccine, now having had, so, and I know there's a lot of debate about this, but if having had COVID, getting re-vaccinated, or in fact, if I got the placebo, vaccinated for the first time, in fact, is a good thing for me to do, and will continue to ensure, you know, a certain degree of health and resistance to the disease, I should know that, and I think it should be required of the trials. So if the Janssen vaccine gets approved, then I think they should unblind the trial for any trial member.
So you can request that. I mean, many people have requested that, and
yeah, no, I will.
It unfortunately doesn't help with our data, but you can request that. And you know, I mean, if
I am a little bit more interested in my personal health than Janssen's data, although I understand how important it is to have good data.
Ultimately, the other thing is that you can get a virus, you can have an antibody titer, and then you can actually determine how protected you are currently based on your antibody level. And if your antibody level is not sufficient or it really should be higher, then you can just go ahead and get the Moderna or the Pfizer vaccine or anything else.
That's a great answer. Thank you very much.
Yeah, and thank you, Richard, for, I asked Richard to share information on the trial, and I also applied, and then I sent it to a few friends, and I was rejected for the study, but a number of friends have been accepted. So thank you, Richard.
Yeah, this is extremely valuable. And, you know, unfortunately, they when you sign the consent form, it should, you know, describe all these stipulations that are related to the trial.
No, I know that, although I'll tell you that the medical research facility that I went to when I got the trial vaccine told me that all the major pharmas had agreed, if their vaccines were approved, that they would, at the trial participant's request, unblind the trial. So you should, and I actually think that that's a kind of fundamental patient right? That you should be able to know whether you got the placebo or the actual vaccine.
Yeah, exactly.
I have a question about you just mentioned that you recommended him he could get an antibody titer. I've been wondering myself whether, you know, I've had my first shot, I'm waiting for my second after that supposedly kicked in. I'm sort of wondering if I should get an antibody titer at some point just to make sure it worked with me, or if I ought to do it 6 months from now and see if it's still working.
So you always can. I mean, I whenever it's going to be available from the healthcare provider, of course, you always can. Yeah.
What is it called again?
Antibody titer.
How long can you get that since exposure? Like, how long do you have where it can be detected?
Well, no, I mean, so if you get, like, for instance, the Pfizer or the Moderna vaccine, and you have to wait until you will get both shots. So you will have the maximum effect that vaccine intended to give you after 2 weeks after the second shot. So you definitely have to wait until then. And then you can get a titer then. You can wait a year later just for out of curiosity and see how does that work at that time.
Okay.
Ilkiko, what are the best statistics now showing about how long you have the antibodies after you've had the virus? I think that's one of the issues.
That's the thing that we don't know yet. So I mean, all these trials for all the companies are going to continue. So Johnson & Johnson has a trial ongoing for 2 shots. So we evaluated the first, we determined that it's actually sufficient, but we actually continue another trial where we actually use 2 shots just to see if it is even giving any higher or longer term protection.
But if you had the disease, and we now have people in this country who have had it back in March who have recovered, right? How do we have good numbers on when your antibodies disappear from your system?
Yeah, we don't. But again, it's so new disease that we just don't have more information. I mean,
okay.
It is only a year long so far. Yeah.
We're almost out of time. Maybe time for one more question.
So when will 19.2 come on?
I'm sorry?
When will COVID 19.2? When do you expect that to show up?
Well, unfortunately, it is circling around. So that is why it is so incredibly important that once the vaccines are available, all of us should get vaccinated as fast as possible. We are really running the clock against the mutations. So the faster we will be able to reach herd immunity, the better it is.
Thank you.
We actually cannot, we don't want to give a chance to this virus to keep mutating and keep infecting people.
Does anybody know, is it the number of vaccines available that has created the tier system for who's eligible?
I guess, yeah, I don't know how many total stockpile there was or there is currently in the US. I don't know. Yeah.
Okay.
I have a question. Right back at the beginning, you said something like the virus isn't alive until it gets into a body and then it starts to replicate.
Yeah.
Does that mean that viruses can be thousands of years old?
Yes.
They can be. Yeah, okay, thank you.
And they can be in the earth, or they can live on surfaces, and they can be there forever.
Okay, thank you. So,
can I ask one question? Can you hear me? This is Julie.
Hi, Julie.
Hi, I'm driving. Sorry. And so it makes sense to me then, you know, the why we keep wearing masks, just for all the reasons you just said, Ilkiko, right? Because variants and all of that. So, I mean, this is a hard question, but do you foresee a day when masks aren't?
So once the herd immunity is reached, that's really the time. And when we will declare victory, that like 70% of the population is immune either by vaccination or by getting the disease, I am not sure when we are going to reach that, but hopefully the CDC or some health authorities will give us the thumb up.
Once we get the vaccine, can we be around people who have not been vaccinated?
You can, but you have to wear your masks and you have to distance yourself the same way. And one of the reasons is that you actually could get again the virus, of course, right? So the virus again come to the body as you saw in the video, but you will be able to attack that. However, during that time, especially maybe the first couple of days when you are reinfected with the virus, you are also going to shed it. So when you sneeze or when you speak, then you actually can infect other people. So that's why it is actually very important that we all have to continue to wear masks and have the social distancing until the entire population is reaching the herd immunity.
Well, even in countries like Taiwan, where they haven't had a case of coronavirus for, you know, over 300, 400 days, everyone wears a mask outside. I mean, they still, everyone just, that's just the way of life. Everybody wears a mask. So, you know, yeah, I mean, I don't think they don't, you know, it's not sort of an indoor thing where you have to, you know, distance yourself from friends and, you know, friends and family when you're inside. But when you go outside, it's just protocol to wear a mask. So it, you know, it could be that we end up in that situation too, which is not such a bad thing, you know, if we stay healthy and we're alive.
Yeah.
You know?
I do know Pete did put a question in the chat. Why are the number of vaccines being developed so much greater than the number of treatments? I don't know if you know that.
So actually, the number of treatments are pretty high too. I mean, almost every compound that has many compounds which are normally used for immunological diseases have been actually tested and repurposed for treating COVID-19 patients. There is a very high number of them.
Well, thank you so much. I think if it's okay, maybe if people still have questions, would you be okay with them?
Sure, you can email me, you can ask me. I mean, I am happy to answer it if I know the answer, and if I don't, as I said, I mean, I either can figure it out, or I can suggest you to talk to your medical professionals or medical doctors.
Thank you so much.
Thank you. Yes.
Thank you, Ildiko. Thank you, Rebecca.
Thank you.
I hope it was helpful.
Very helpful.
Yes, very much. Thank you for taking your time with us and answering all these great questions. And it's such a blessing. So we're glad you're at St. James.
Rebecca, can you stay on? Can you stay on for a moment?
Yeah, I will.
And Claire Friedman and Bruce Ray, can you guys stay on too? I have a personal agenda item, which
I'm gonna head out. Thanks, everybody.
Bye.
Hey, Jane.
Hey.
Thank
Good
you,
to see you
Ildiko.
all.
Thank you, Ildiko.
We're talking about poetry.
We're talking about poetry and performance. So did Claire stay on? Did we lose her?
No, she's jumped off.
Oh shoot. So Claire is now, we now have our 6, well, assuming that Laura Hargreave is also going to read, which Walter was almost certain that she would. We've got our 6 readers for the Ash Wednesday, and we're all set up, and Walter and I have kind of become co-producers of this. Walter's very enthusiastic about it. He's going to be one of our readers, and we're gonna record it and hopefully get and you know, Walter's going to try and mix it and see if we can't get a